in all of these ways. So that's a regular way it goes for techniques and so on. We have just finished with the screening procedures and the screening possibilities for the different compounds, and have explained why we are focusing on the drug discovery processes in the preclinical development area in this way, and why we are focusing there on small compounds, small molecules in this way. What I have not fully explained is in which way we are focusing on some diseases. There are some social burdens in this way, and some financial aspects which are important for the different aspects. But we just spoke that some people really focusing on cancer, for example, for the different purposes. Here you see that cancer is one of the most important reasons for dying. There's very high social impact for cancer, and that cancer is quite important as a disease form and life-threatening disease form, reducing life quality, and so on and so on. So there's a big burden in this way. On the other hand, you can see that smoking, alcohol, and diet, or missing diet, are also some of the main reasons for cancer, and that cancer can be just be beaten just by inhibiting these areas. And you see that in India, many of these areas are really followed up, and that should lead to a lower number of cancer aspects in this way. On the other hand, you see here, cancer as a disease is an important factor, but if you then look into more detail, you can see that we do not have only one form of cancer, we have many different forms of cancer. And if you look here for breast cancer, colorectal cancer, or endometrial cancer, these are cancers which are localized or on their tissues. Sorry. If you make the characterization by the behavior on cell modification, then you have even a different modification of cancer, or a different translation into different cancer cell lines. So some of the breast cancer cell lines could be related to colorectal cancer because they have the same kinase motivation and things like this. So it is not really sufficient to just focus on cancer, which kind of cancer you are focusing, and on which target you are focusing in this way. So that is much more than say, I'm just working on cancer also, or just I'm working on neurodegenerative diseases. You see the disease selection, which have local centers worldwide, and centers with emphasis on a therapeutic area. And you see here that cancer, psychiatric or neurodegenerative diseases, and infection, they are the main three of these different areas and so on. So these are three high defined disease forms with a high social need, really, and with a high medical need. So there is some topic in this way. But you see here also dermatological, ophthalmological, orphan diseases, and so on. They even have some points. And I will come back on Friday to orphan disease, and you see here orphan disease with 35% are quite often. And orphan disease are rare diseases in this way. So the main problem is here that even rare disease are rare, but you have many of these rare diseases and so on. And thereby, if you have so many diseases, then you have so many different forms which can be formed. As you have more than 8,000 different rare diseases, then the total number, if you put this together, are quite large. But if you focus on one of the rare diseases, it's difficult to find patients because they are so rare. So that's the reason why we are looking on the different areas. And you see here human targets like enzymes, catalytic receptors, transporters, G protein coupled receptors, different growth hormones, and so on, and even sometimes other proteins are really accepted here. So there are different possibilities in which way you characterize your target. You characterize by disease, by a specialized disease. You're characterized by a molecular target in a way, or in which way some synthetic classes, so anorganic antibodies, endogenous peptides, metabolites, and so on. So there are also different classes in which way you can describe your target for selection, for further developing, and so on. So mostly you see here synthetics are the one we are describing and prepare, and we have in our synthetic route for these compounds in many of these ways. What I would like to emphasize here also, that there are some tendency, some trends in medicine chemistry. One of the trends, the last 2 decades about this, is to work on covalent drugs, drugs that bind covalently to the target, and thereby, in most cases, make some inhibition, and this inhibition had then destroying the target because it's covalently bound, and then you can see here. And then you can see here that this development here in different compound classes are really going on. But on the other hand, you must see if the target is covalently bound, maybe it's not only selectively bound. And when it's covalently bound to a non-specific receptor, then your toxicity is tremendously increasing. So thereby, although this kind of targeting is very promising, it takes simultaneously high risk of unwanted side effects because of the mechanism of action of this different structure. And thereby you must be aware that when you develop compounds, then with a compound like covalent drugs, you cannot only focus on your target, you must also focus on off targets, which can then be applied for the different structures. So thereby this strongly affects the different aspects in this way. Yeah? And you see here with new chemical developments, you see here antibiotics in a major field, more than 50% of this newly approved drugs and so on. With the antibiotics, we do have a very specific problem. Because if you develop a new mechanism of action with the antibiotics, it's very important, very new, with very high use for society and so on, because when you cannot treat the infection by any other antibiotic, you take your new antibiotic in this way. But it also means that your new antibiotic is only taken when the others are not working. So it's a reserve antibiotic. When you have a reserve antibiotic which is taken very rarely, then the prescription of this drug is also very rarely, and you earn money very rarely in this way. Yeah? So the development of a new drug in the antibiotics is of high social burden, high social interest in this way, but from a pharmaceutical industry who want to earn money, very difficult to take on. Because as long as it is a reserve antibiotics, it stays very reserved, very rare in this way, and thereby you get only very small amount of money in this way. And this for a longer time, so if this compound is such a specialized mechanism of action, it keeps it antibiotic resistance or property for a very long time, then it maintains this spare antibiotic for a long time, and then the generics get off because your patent is running off. So that's really difficult for pharmaceutical companies to develop really new antibiotics because they want to earn money, and there one has to think about other revenue sharement in this way, in which way this is really attractive for a pharmaceutical company. But that's also a point where the academics stay out, because we are not really dependent on this income for the money. We are really on the responsible that we pay back the society our money. So thereby antibiotics is quite often be researched and be developed new mechanism of action by the academics in this way. So that's where you can see that you do have different possibilities and different properties where academic may be a better way, a better place for research than even pharmaceutical companies in different aspects where we're looking for. Yeah? And you see here from the newly approved drugs, you can see here oncology, metabolic and immunological diseases. They have one main point in this way, because here you have a threatening of the patient, and you can earn a lot of money. This is what is really where you get the money from the pharmaceutical industry. very small amount of anti-infective, and then neurological, and also rare diseases, because that begins up to be attractive because of different new modes of the administrative regulations, that you get higher protection and better payout for rare diseases than for any others, because thereby you help these patients for a different thing. Yeah? Nevertheless, you can see that the number of new approvals, new FIC means first in class, so new compounds, the green one are new first in class, the others are me too or some other opportunities, whereas we have a drawback in 2016. Now it comes back slightly to a larger amount of new chemical entities going into this way. Yeah? You can see that the differences here are really coming out in the different areas. And what I would like to stress here, that there are different possibilities for getting marketing approval by the different drugs and getting through the different clinical trials. Yeah? You can see that one of your compound can be named as breakthrough therapy, which means a new mechanism of action, or causal treatment where before only symptomatic treatment would be useful. So this breakthrough therapy in normal ways got a fast track approval. Fast track approval means that you get faster inspection by the government, and that you get reduced number of clinical trials before approval in this way. Yeah? It has been done by some Alzheimer medications, which were only partially successful in the way. So they were treated as breakthrough therapy. They had a fast-track approval in this way, and they have been approved in the United States with limited data source and so on, and they were not approved in Europe. So that, yeah? And you can see here, priority review, accelerated approval. This always needs some additional approval where you can shorten up your time getting into market. And since your patent time is limited, and when you get earlier into market, you have more possibilities to earn some money for the compound. So that's really essential. And when you have a blockbuster, really, and this blockbuster makes $1 billion, really, or something like this, then it's really essential to be 1 year in advance or 1 year before this. And then you see here some specialized programs, qualified infectious disease program designation, or orphan drugs. And there are some special programs where you get some benefit if you develop some antibiotics, or if you get some benefit if you develop some orphan drugs. Because then you are getting into lower amount of revenues, of lower amount for repayment of your investment, but you get a higher amount of your impact into social health, yeah? Into all of these areas. Yeah? And then at the end, you want to have some first-in-class compounds with these new drugs in this way. Yeah? And this is just that you see oncology, infection, and CNS. These are the main percentage for approvals, whereas CNS, I have to say, is quite difficult, and many companies went out of CNS because they have so many failures in the CNS drugs because of toxicity or missing activity in this way, and that was the reason many companies went out of CNS explorations and so on. Yeah? So thereby, we're going on. What we always have into mind that when we make some drug development, the drug development is target validation, hit generation, lead optimization, and biological validation, and then into preclinical and toxicological screenings and so forth. That's where we are involved, and we can see when we have here a success rate of about 35%, when we have here hit generation, when we have about 200 targets, then not all of the targets are really successful. The same is true when we investigate different compounds. Then even the different compounds, they have a minor hit rate of less than 30% in most of these cases. So most of our compounds are inactive. Most of these targets are not the best target for choosing. And thereby, you have to optimize your target and also your compound step by step in this drug development program in this way. Yeah? And you can see here at the beginning, this is low throughput with high novelty. You have a success rate of about 2 compounds, but you spend about 0.7 billion euro for these 2 compounds in the development, so a very large investment. And at the end, this investment had to come out. So these 2 compounds for pharmaceutical companies must make at least 8 to 9 billion euro or dollar earnings just to be on the correct side, not to make the great investment for the future and so on, but to be on the right side. And that we always have to calculate when we are looking on the income of pharmaceutical companies or what costs are really there. And I always read, the preparation of this cost of this drug costs only 50 cents, and they are selling this for $50 and so on. Yeah? So they are immoral. They do not pay, or we pay too much for this medication. But if you take into account the development line and the cost of development line, and take this into the cost of the drug manufacturing, then it's totally different view. Yeah? So thereby, we must be aware that this is really clearly seen on the different areas. So the different agents, the different targets, and the different diseases, they really contribute to what we are selecting in this way. And then we can make some optimization in which way these are visualized, or in which way they have some overlap with some other compounds. We can inspect the target at the different phases of development in the preclinical and also the clinical development. And then we can see if our cell target is really successful, if we look for a good animal model, and if this animal model gives good transformation into the human disease. And you must be aware, a model is only a model. It's not the correct disease. So you have something different in your hands for the different structures. And then the better you have the overview of your disease, the better you have seen in which way it is originated, in which way it is progressing, in which way it can be beaten, then the better you can describe your compounds and then go into further drug ligand investigation and can select these compounds in this way. With this, I would go 3 or 4 steps back. So we start now once more with the very first beginning, and you see here I'm just with the elements of the humans, and thereby we can see from the elements we build up some molecules, and from the molecules we go then into drugs and into different other areas into this way. So thereby it's the elements which makes us, and then we go for the next step into this way. And then you can see how many molecules can be made. So, and I believe that the number of molecules which can be made are as many as the stars in our universe. So thereby we go from the very small to the very big of the different possibilities in this way. And thereby you can see a 10 high 40 or 10 high 120 compounds can be prepared, and this is a huge amount, and thereby not all thinkable compounds should be prepared. We should focus on some of these compounds, and focus is then, if we look on a library of compounds, if we look on the chemical space, what is fulfilled with the chemical space of the different compounds, and in which way we can focus this compound. Can we focus this on larger compounds, or can we maybe focus this on fragments? And when we have very small fragments, later on we can put on these fragments together and then have a larger molecule which then have the optimal properties in this way. So thereby, what is our strategy? Do we look for the whole molecule? Do we look for active fragments into the way? Different strategies we have to decide we're going on, and we can take all different strategies. So you will become specialists in one or the other property in this way, and so on and so on. Many of this strategy depends on your screening behavior, on your screening assays. So can you screen for fragments? Maybe you can make some fluor NMR displacement on your target that's possible, and then you can easily screen on fragments with NMR studies and so on. So there are different possibilities. But if you do not have a fluoro-based compound for displacement, you have some difficulties in these kind of screening. So thereby, your screening method also have to be included here into your development of the compound and into the development of your chemical space within this area. We have been focusing, and I already showed you this slide at least to a certain extent, we have been focusing on aminergic receptor, thereby we are focusing on histamine and dopamine receptor. We have a certain space where we're looking at, which is convinced by this area, and there we try to make our chemistry what we are capable of doing, mostly to expert in these fields. Yeah? Some catechol, some heterocyclic chemistry, small molecule chemistry, mostly hydrophilic chemistry, because the biogenic amines are hydrophilic ones, and thereby we can easily go into the different aspects for the compound. And then we develop this chemistry and apply this chemistry also for other structures. Just one example, we made some very nice thiazole compound as a heterocyclic derivative, and then with this thiazole chemistry we were very familiar. We took this over into the dopamine field, where we also made some thiazole derivatives in this way. So we are not restricted in chemistry to the certain fields. We expand our chemistry then to different fields which are nearby into the different areas into this way. Yeah? The first step is in many cases trial and error. We make a compound, test the compound, and have a failure. We make a new compound, test this compound, have another failure, and so on. And that we not have 3 years of failures. There, in most cases, we must need some lead structure, some endogenous ligands, some structure where we can work on, and then make some hit generation and lead generation for the different structure. Yeah? This can be done by rational design. If we know the target, then we can identify the binding pocket, and then we can, by computational approaches, prepare some compounds on this binding behavior. We make some virtual screening on these different compounds, and then even sometimes we do not have to prepare these compounds. In many cases, you can buy these compounds. And buying a small library of compounds is in many cases much cheaper than preparing them on yourself. So there are large libraries which are available, and then you can buy some compound and take them for screening. At the end, you still have to synthesize many compounds, because not all of the libraries fulfill all of your substitution criteria, variation criteria, and so on. But in many cases, the buying of library is quite a good starting point for many of these things. Yeah? And another point, which was highly topical 10 years ago, combinatorial chemistry. Because you put 3 or 4 compounds together and then make a large library of different compounds, and thereby have, in a very short time, a large variation of different compounds with the same scaffolds, but in different combination areas in this way. So if you go for optimization, I think it's a fine approach. But if you go for hit finding, it's also a trial and error, more or less. But there are also possibilities in the way. So the combinatorial approach is a different approach, but with huge possibilities. And if you have 1 or 2 very highly potent scaffolds, then you can go into new scaffolds or new scaffold arrangements with combinatorial chemistry, which makes really sense, and you can easily proceed with some of these compounds in the structural elements. Yeah? Within the last decade, but especially within the last 2 years, artificial intelligence becomes more and more pressure in these different ways where we can go on. And whereas ChatGPT and things like this, they recognize pattern. Yeah? You see, molecules, binding molecules exist of pattern, and thereby this pattern recognition molecule must be of high use for medicine chemistry. There are already some applications, and you can see with the AlphaFold or AlphaFold 3, meanwhile, that, do we have 4 already? I'm not sure exactly. But this AlphaFold makes a very high predictivity of 3-dimensional structures of proteins, which was unexpected 5 years ago, and then comes out of the blue. Yeah? Now we have DeepSeek and other recognition patterns which go with small computing power and so on, and I'm pretty sure that we are at the beginning of this generative artificial intelligence at the very first beginning, and I hope that we will stay at this weak artificial intelligence for a while, and that we are not going for strong artificial intelligence, which are much smarter than we are. So that's another point, but you see they're always connecting. And like many people where they say, oh, we are, artificial intelligence is too aggressive and they will take our jobs and so on. I don't think that they take your jobs, but they will be helpful in your jobs. So you have to prepare less compounds. They will be tools that you have to apply, and thereby I strongly believe that you have to work with artificial intelligence, and we are actually developing different tools for this development areas in this way, and you should be aware that you are using these tools in this way. I'm also aware that these tools are not really user-friendly at the moment, but I'm pretty sure that in half a year or maybe 2 years, they will be much more user-friendly, but I'm not sure if you have to pay for these things or not. Yeah? I'm pretty sure the companies will make you pay for these things in one or the other thing. Yeah? There are many tools available where you can freely test your compounds, but be aware of the copyright condition of these freeware tools. Sometimes when you put in your compound you want to test, you say, with giving this to this company for measuring lipophilicity or something like this, or calculating lipophilicity, you're making this open access. You're losing the possibility for patenting or for protection and so on. And if you give one compound, then the related ones are also non-obvious, and you're losing your patentability of your structure. So be aware of the copyrights that's connected with these free tools, especially these free tools in this way. So sometimes it's cheaper to spend money on commercial tools than to use free tools. Yeah? So cheaper in a good way for the different structures. Yeah? Nevertheless, when we go into compound development, you see that when we start with the high throughput screening, we have some hits, we have some activities, lead and drug candidates, and you see we easily go from 1 million to 2,000 to a few hundred, and then at least we end, hopefully we end with one drug candidate or with drug. Yeah? So when we have one drug, then we can earn really money and get into the different areas. These different compounds with these failures, they have been plotted here. And we have here the one dot for each compound in this area. And you can see here, we on the x-axis, the molecular weight. And you can see here the lipophilicity of the compound. And you can see that we do have a hot point here, yeah, where many more compounds have a medium molecular weight between 300 and 500, maybe 600, and have a log p value between 2 and 5 or 6 about this. So we can already see that some preferred compounds have a good molecular weight of about maximum 500 or a little bit less, and have a lipophilicity between log p values between 3 and 5 or something like this. But we also see that there are some compounds which are really outliers on this, so they have a totally different molecular weight, totally different lipophilicity. Yeah, so it could be, it's just a rough orientation, a thumb rule, that these are beneficial physicochemical properties of the compounds, but not necessary for the compound. Yeah, so thereby we go for the different structures. And you can see here, safety, solubility is strictly connected to lipophilicity, is connected to permeability, is connected to CNS bioavailability, is connected to metabolic stability, and also to target affinity. So you see lipophilicity is a central aspect we connected to different of these areas, and we can really introduce lipophilicity more or less into the different compounds into this way. Yeah? Thereby, we can see that the physicochemical description, like dissociation factor or lipophilicity or electronic parameters, which are more hydrophilicity parameters, can be easily described by mathematical models, by computational models, by molecular modeling and things like this. But these are models also, yeah? And at the end you have to experimentally calculate or check your compounds on these physicochemical properties. To show the influence of these physicochemical properties on pharmacokinetics, for example, you see it includes here lipophilicity influences the absorption, influences the distribution in the different areas, distribution in different chambers into different physiological behaviors in this way, and then with the lipophilicity you can have different shapes. So lipophilicity does not mean solubility. There are different aspects in this way, and depending on the hydrophilicity, depending on the lipophilicity, depending on the polar surface area, your compound is more or less lipophilic or so on. So that strongly changes in the way. And we can also see that hydrogen bonding or ionization also changes with the structure. So if you have a basic compound and go into an acidic compartment, it would be protonated and totally change its physicochemical properties. So that's quite important that you look on the basic physicochemical properties. The basic physicochemical properties may be connected to absorption or distribution and connected to your target, for example. One example is when you have non-steroidal antiaromatics, yeah? Cyclooxygenase inhibitors, COX inhibitors, like acetylsalicylic acid. Yeah? These are acidic compounds, ibuprofen, diclofenac, acetylsalicylic acid. So they are easily soluble at a pH of 7. But when you go into inflammatory tissues, these tissues are more acidic because of the inflammatory processes, and thereby your compounds stay with they are less soluble, and then they stay with a less basic compartment, and then you have an enrichment of your compound in this inflamed tissue, which gives a higher activity of your anti-inflammatory drugs in this tissue. Yeah? Higher COX inhibition by these drugs, by these physicochemical properties. Yeah? And you see paracetamol, another non-inflammatory drug, has not this acidic compounds. It is much less effective. Yeah? So connected to the physicochemical properties and the COX inhibition into this way. So thereby, we have to investigate the solubility of the compound. Why is solubility the first aspect you have to investigate? Do you know why solubility is so important for this compound? Why? For blood system, but the first and important issue here is when you make the screening in your buffer, in your test system, it must be soluble. Otherwise, you cannot determine the concentration. So even before you go into blood or absorption and so on, solubility is one of the essential elements when you go with a compound to the pharmacologist. Yeah? The first question is, how soluble is this compound? Because otherwise, he cannot test this compound because the concentration is not really clear if you go for the different aspects. So solubility, quite important. And then solubility often determines lipophilicity, or this determines different aspects, or this has to be calculated, determined. I prefer experimental determination of this structure. Permeability, in which way it goes to cells or other structural elements. For example, if you go for CNS compounds, then the blood-brain barrier is a quite important barrier, and then you have to check in which way my compound is crossing cellular membranes into this way. And there are different models in which way you can go for this different structure. The hydrogen bonding and the acidity or basicity, like a pKa value, are also quite important because this determines the protonation or deprotonation status of a compound. And you see here some description in which way you determine these data. I can go into detail when you have some questions on this. Otherwise, I have so many topics I have to discuss with you. I will go through and just mention a few of them into the different aspects. So please interrupt me when you have some question. No problem at all. And I would be fanatic really to answer these questions, because I think all of these aspects are really important. But due to lack of time, I would just skip some of the details described here. The detail I would not skip here is the structure physical-chemical properties relationship. So the SPR relationship, not the structure-activity relationship, but structure physical-chemical properties relationship. The SPR is quite important for the structure. So sometimes I do have some compounds with a moderate or good lipophilicity, yeah? But when these compounds are crystallized and they are flat, then they lay one on the other, and they do not have any solubility really anymore. So they do have appropriate lipophilicity, but they do not have exact solubility after crystallization. If you keep them soluble, then it's okay. But if you want to make them soluble from a crystal, it's not really working in this way. And then you have to go from these flatlands where you have the PP or pi-pi interactions, then you have to make some compounds with some other structural elements which avoid this flatland interactions, and then you can easily go out of this. But you must detect these problems. Yeah? So you can easily go over some of these problems related to this, but you must be aware that they are existing in this way. And that's really a problem into the different areas. Yeah? And you see here we have different problems: greasy, not large, not greasy, large, not greasy, and large and greasy. They have the highest problems in this way and have a lowest scoring in the possibility for development, whereas you see where we have small molecules, so not large, and have good solubility, then your success rate here means high or medium developability is quite large for these compounds. So keep into mind that your compound should have a moderate activity or moderate size and moderate thing. I have to apologize, but the golden way in between is the best. Not too hydrophilic, not too lipophilic. Yeah? Because you have to be aware that your compound must be water soluble. Our body exists mostly of water, so it must be water soluble. But it must cross biological membranes, and biological membranes are mostly cells. Cells have the lipid biolayer, and the lipid biolayer is strongly lipidic. To cross this by passive diffusion must have enough lipophilicity in this way. So hydrophilicity enough to be water soluble, lipophil enough to cross the barrier of cells and some other area. So that means, and that you can achieve with low molecular weight. Maybe I stress also one other point. If you go for fragment design. Fragment design, you have maybe 3 or 4 or 6 different fragments which are more potent in this way. And then you add one fragment to the other to have 2 fragments. 3rd fragment, 4th fragment, and 5th fragment. And then your compound goes larger and larger. Mostly goes more lipophilic and more lipophilic. So maybe you increase your affinity, you increase your potency, but your lipophilicity and your molecular weight is also steadily increasing and giving lower chance of being developed. So try to stay with the smallest lead structure as possible and make then the optimization. That's the general outcome we're working in. Here you see the importance of aqueous solubility, and I already stressed there are some classifications due to the FDH and the EMA, the European Drug Agency, and also all drug agency. They have some nomenclature on solubility, which is freely soluble or poorly soluble or slightly soluble in this way in mg per milliliter into the different ways. Yeah? And then you can hear your compound dissolved in DMSO, and then you put this in an aqueous buffer, and then you mix this, and then you read here the fluorescence or something like this, and then you measure the solubility of this compound, because the soluble compound has a different fluorescence than the particle structure in this way. So thereby, you can easily check the solubility in a way. One point here is that in most cases, you put some energy here into this structure. So it is an energy-driven solubility. So if you maintain this for a while, maybe you will get a larger number of the molecules stuck into the ground and have less solubility. So this is a kinetic solubility where you put energy into this and maybe over-soluble some solution, where when the energy gets lost time by time, then your solubility gets reduced in different ways. Yeah? So must be aware. And you must also be aware if your compound was dissolved in DMSO, then you still have a small amount of DMSO included into this way. Yeah? So must be calculated within the different structures in this way. So this preparation here is one possibility to make the kinetic solubility motion, but there are also the thermodynamic solution where you have your compound and then add your buffer into this area and then shake it for a while and then leave it for a certain extent and then measure the dissoluted area here by UV or by MS, for example, measure the concentration left in the amount you have. Yeah? Very difficult method. You have to be exact with all of the amount you're giving there, but very exact measurement into the way. Yeah? And you see when you want to give, for example, your compound to pharmaceutical industry for screening and so on, yeah? Some information on solubility is always needed. Yeah? So that's really in many areas. The other point is that the solubility is the factor for screening. Then for interaction with the target, we have the lipophilicity as the other target. Yeah? So solubility, dissolution is different, and then we go into lipophilicity. Lipophilicity can be easily measured by log P values, where the octanol water dissolution in this way, and then you can measure the concentration in one and the other compartment, and then you can determine the log P value very easily. Yeah? Nevertheless, if I give this to my PhD students, they are always fed up with this because it's a long time doing this. You have to be very exact. You have to repeat it several times in this way, and the information is also limited because our body system is not octanol water. The water is at a pH of 7.4. So the 7.4 pH value is much more important for blood vessel and blood tissue and so on. So then the log D value, where you have water which is with a correct osmotic value and mostly also with the correct pH value, and then you have the solute here with the octanol water at a pH of 7.4, could be more helpful into this way. But then you can determine both of them in this way, and you have double the work. Yeah? So thereby you see even the easy the determination of log P value or log D value gives a lot of work into this way, but gives a lot of information also for this. Yeah? So thereby you can see in the different areas. And you can see here octanol water phase, log D value, where you have here, for example, the protonation or deprotonation status at a pH of 7. This gives much more impetus. Is it monoprotonated, deprotonated, or is it any other interaction with the phosphate buffer or something like this? So there are different possibilities you're going. To make it then going into detail and to show why we are looking for log P and log D values into the different areas is here that you can have different interaction with your biochemical assay on the potency, depending on your lipophilicity. So your compound, for example, could be diluted into some lipophilic pockets. So normally, with an increase in lipophilicity, most proteins, they have some lipophilicity pockets in the way, and with the increase in lipophilicity, you increase your binding affinity in many cases. Yeah? But that's not helpful because it's just a lipophilicity gain and not any other thing in this way. Yeah? And it's also possible that you have some other lipophilic areas, not on the molecule, but in the cell assay and so on, and that your compound stuck to some other assay ingredients and then deteriorate your concentration you're having there, and then giving lower or sometimes also higher concentration because of the differences in the distribution for the area. And thereby, this is a direct effect on different targets and so on. Here, one example is the HERC activity. HERC is a human ether-related agogo receptor. It's a potassium channel. It's a voltage-dependent potassium channel in your heart. And it controls the rhythm of your heart, the electronic rhythm. And if that cardiac channel is inhibited, you got rhythm distortions and so-called torsade de Pons, rhythm stopping in this area, arrhythmic behavior, and then you die. So very important toxicological aspect here, always tested with drug development before going into humans, the HERC channel in this way. And you see HERC channel has some lipophilic pockets, destroying your binding area in this way. And the same is then with different pH values and different protein binding. And even when you have just your cell assays, with your cell assays you have some other proteins, and your compounds could have some reversible protein binding properties, and then it's taken out of the equation for the concentration there. And then you have a lower concentration in your structure, which gives some effects on the potency, and you detect a lower potency of a compound, although it is much more potent maybe in real estate. Yeah? So this is a direct effect that could be connected to lipophilicity in the different areas. You see there are many lists of compounds where you can detect lipophilicity or the influence of lipophilicity, here delta log D value, and the expected lipophilicity potency in this way, in which way this is gaining for lipophilicity in comparison to proton. For example, when you exchange a proton to a methyl group, a phenyl group, a chloro substituents, or some different alkyl substituents in different ways. I do not expect that you select or learn these lists in any way, but I think this list here from a publication and also from the drug hunter list in a way, which is freely available. And then you can go into details, and when you find, for example, that your compound, your chloro compound is nice, but your bromo compound is nicer for activity, maybe you're not going then for yodo, which may be also helpful, but you see that this gain in activity or loss in properties then is just by lipophilicity for the structure, and then you go for another different structure. Maybe you go for a trifluoromethyl group, which is metabolic then more stable and has better quality in the different structures and has different aspects into this area. So thereby, the selection of the substituents are quite important. And here you have a list in which way these contribute more or less to different activities in this way. And you can see here amides, reverse amides, and different possibilities, sulfonides and sulfoxides, piperazine molecules, and so on. So thereby you can choose different compounds in which way you gain or lose lipophilicity in the different areas. And you see here with the basic moieties or with the oxygen ether moieties, you got a loss, you got a higher hydrophilicity for these compounds, so they are much more soluble also in many aspects. My main point when I discuss with my PhD students some drug optimization process is to look for such behavior. Because they can prepare maybe 100 compounds with chloro, fluoro, bromo, or yodo substituents, or dichloro, dibromo, diodo substituents, and then make all of the different variations, and then they have at least 100 compounds which are very nice and they have a very clear structure-activity relationship, but it was more or less obvious when you had the first 10, and then you made the other 90 also in this way. Yeah? The main problem with us is that we are limited in our screening resources, and thereby we go for quality and not for quantity. So in which way we can optimize these compounds for the lowest number of variation and the optimal number for pharmacological activity in this way. And pharmacological activity is then connected here to lipophilicity and so on, and thereby we do not test all of the different structures, we test a small number of these different structures, and thereby limit the variations we are doing. Yeah? I must confess that sometimes this is frustrating for the PhD students to do this, because not all of these substituents are commercially available, and sometimes just changing the position from one to another takes 3 months of preparation, and at the end it's just auto and meta or something like this. So that's frustrating, I see, but nevertheless we are going for these variations and to the smallest number of variations to check in which way these compounds are really accessible. Yeah? And I find this mostly interesting for heterocycles, because we are really often working for heterocyclic chemistry, and here you see the changes if you change from a phenyl to a diazine or a 5-ring membered ring system, and you see maybe sometimes not so expected here. Here's the combination or the comparison to hydrogen or to phenyl. So in parentheses you have the comparison to phenyl, and you see with the thiazole moiety it is less lipophilic than the phenyl, but more lipophilic than the hydrogen moiety in this way, and then you have something in between for the different structure. And then there are also possibilities in which position you're changing this thiazole moiety, so there are also some other possibilities in which these are really gaining in this way. But very often it is better not to make all of the optimization of the phenyl with a substitution, but to exchange the whole phenyl into another heterocycle, to 6-membered or 5-membered aromatic heterocycle, maintaining your PP interaction, but having different lipophilicity. And when you then go for heterocycles with the nitrogen, oxygen, or sulfur, then you have also the different possibility for hydrogen bonding or hydrogen acceptor possibilities with a different structure, getting more increase or more insight into the different interaction. So the heterocycle exchange is in most cases much more interesting than substitution on the phenyl structure. Yeah? You will see later on that not always successful in many cases, but very often helpful in these different areas. Yeah? So thereby you can easily change and detect in which way these reactions could really then be followed up in the different ways and the different areas for lipophilicity and hydrophilicity. When I showed up with these slides, I had some ideas in which way I discussed different aspects in detail or not in detail and so on. And then I found one slide where there's a common observation, for example, that you have increased lipophilicity with a terzabutyl or phenyl group, but then some other problems occur in the different areas and so on, and there are different possibilities. You see this from also from Drug Hunter.com, yeah? So this is freely available where you can get this discussion around, and then you can see that the phenyl moiety very often gives some problem with cytochrome P450. And then you need with the phenyl some substitution or some exchange. Maybe some stronger problems with the heterocycles than the phenyl moiety or things like that. So thereby, maybe you have to do in the para position a chlorosubstituent or a fluorosubstituent, then to avoid cytochrome P150 interaction and to make the compound more stable. It increases lipophilicity and maybe changes your CNS penetration with the compound. So maybe not beneficial for activity, but maybe beneficial for metabolic stability. And thereby, you have to calibrate your optimization procedures, what is best and what is worst in this way. And that's why I so much put the importance understanding of your target. If you work on dopamine receptor, what we are doing, dopamine receptors are in the periphery and in the CNS. we mostly want to address our compounds in the that means we have to get in rather high lipophilic compound, which log D or log P values between 3 and 5, because then we have a good blood-brain ratio, because they are higher in the brain than in the blood, because our brain consists of many lipids, and thereby the slightly higher lipophilicity is useful for this. But we do not want to address the peripheral dopamine receptors, and thereby the higher blood-brain penetration area for the brain is beneficial to have a lower activity in the periphery, but higher activity in the CNS. Yeah? And then we want to avoid to have active metabolites, because active metabolites in most cases mean more hydrophilic. Active metabolites with more hydrophilicity means also more in the periphery, less in the CNS. And then metabolize our compounds to have less metabolic structure. So even with the design of the compounds, you have to design in different properties with this different structure. You have to design hydrophilicity, lipophilicity in the compound, hydrogen bonding, acceptor or donator properties in the different areas with a different compound, and then can expect different properties here. Yeah? Here you can see that the amount for the variation here is quite changeable. So if you have a 2 hydrophilic compound, it means it is normally very well permeability, intestinal and also CNS, but then it is quite often low metabolic liability. So these compounds easily go through renal elimination, and then they're getting lost, yeah? Even with direct elimination processes and so on. If you go for high lipophilic compound, then you have low solubility, and you, in many cases, you have higher metabolic liability, because the compounds are in most cases, they are bigger in this way, and you have more possibility for cytochrome interactions and other different metabolic enzymes for the different interactions. The larger your compound is, the more possibilities for interaction you have. The smaller it is, the more limited are these. Yeah? And now you see here with a log D value of 1.3, you have a good range for CNS and low metabolic stability, or 3.5, a little bit lower solubility, but increased metabolic liability. So thereby you have to handle, but between 1 and 5, I think that's a good handleable way for the different structure. Yeah? And that means there are many ways in which you can calculate this lipophilicity or hydrophilicity for the compound, but it's, you can also make some TLC conditions, for example, to make some experimental conditions. Because if you make some calculation by computational approaches, then it goes like a fragment-based addition. So it takes some increments, and your compound is consisting of a number of different increments, and then they are adding these increments, adding some regio-specific factors in this, and then they're calculating the lipophilicity of the compound. And we have very often obtained the calculated one and the experimental one, and they are highly different sometimes, because the increments are maybe then calculated with without pH 7.4, for example. It's just log P values for it. Yeah? And then, so there are many possibilities in which these calculated values not always reflect the real experimental values in many aspects. Yeah? So thereby, then we go for another physicochemical aspect, the acidity or basicity of the compound, the pKa value of your compound, because this is also highly important, in which way then these aspects could be confirmed or not confirmed in this way. And you can see many compounds are phenolic compounds, aminergic compounds, amidine compounds, or what we have seen with the heterocycles, there are aromatic amidines in many of this way, and thereby they have a strong basicity with a compound in this way. And also we have substitution pattern for the different structures, where we have an amine on the heterocycle, and then we do have different structures. So thereby the pKa A value or the log pKa is quite important to have the balance between the protonated and the unprotonated structure, between the acid and the corresponding acid or the corresponding base, or between the base and the corresponding acid in the different areas for the different. So what you have learned in your basic course for chemistry, that must be now applied into medicine chemistry, because protonation status strongly influences lipophilicity. It strongly influences the three-dimensional structure of the compound, because with a new basic structure, a new protonated structure, you have a positive charge, and the positive charge makes other hydrogen bonding than the non-positive charge into this way, the uncharged molecule. So the on-off target potency by salt bridges and so on quite often happens with a different structure. We have a direct interaction with the lipophilicity for the structure. That was the reason why we are normally measuring the normal log P value and the log D value with pH 7.4 in the different aspects. It has aspects on solubility, permeability, and also on the efflux of the different structure. And many of you have heard of the PGP protein. It's P-glycoprotein protein, and this P-glycoprotein is a transporter. It's a ABC family transporter, and this ABC transporter, the PGP, is responsible for the efflux of the different structures. It goes into the membrane, it hits the PGP protein, and before going into the cell, it's already thrown out of the cell. So within the structure, within the cell membrane, it is directly thrown out. And this PGP is, for example, localized in the blood-brain barrier. So it keeps the homeostasis of our brain really effective, because it is efflux of different molecules which not belong into the brain. So it's a protection mechanism of our brain. But it's not the only protection mechanism, and not our own protection mechanism, because some bacteria, they develop some PGP expression. And when you have some bacteria who have some PGP expression, they throw out the antibiotics. So they might develop resistance, because some of these bacteria have a lower and some have a higher expression of PGP. And if you take your antibiotics, then all of the bacteria are destroyed. But they are not immediately destroyed. They are step-by-step destroyed. And if you stop after day 5, for example, then you have a small amount of bacteria remaining. Sometimes your body is able to destroy these small remaining compounds, but sometimes it needs the antibiotic for the additional 2 days, because 7 days are normal intake time in this way. But after 5 days, you feel healthy, and many people do not take then this. And these bacteria remaining, they have a slight advantage of survival. They have a slightly higher expression of PGP, for example. Thereby, they develop resistance. And then this resistant bacteria are surviving and giving this resistance factor to the next generation of bacteria. So for the next generation, you need a higher amount of the antibiotics or different antibiotics, and thereby. So that's one of the reasons resistance is developing, that a small amount of the different bacteria then, with a better survival genetic factors, they get into the thing. And PGP really is one of the point, or this is another name is a multi-drug resistance protein, MDR. So, and the name is also the program here, multi-drug resistant protein, throwing out the different program. We have here called on the metabolism, or the likeness of salt formation, salt stability. And thereby the protonation status is also important for drug formulation. So if you have a sticky, oily base, you can maintain it for a couple of days or maybe some weeks in your hood or in your flask, but then it's oxidized or getting brownish, or it's destroyed sooner or later. But if you have a solid salt form, it's stable over years in most cases. So thereby stability and handleability of your compound, and that after 1 year you do not have to resynthesize, but you give just your salt away, is quite an important factor for drug development. So stability, and if you make high throughput screening, you would not make the compounds all the way new in the synthesis. You have some stable salts, and then make the solution of the stable salt, and then test these compounds. So thereby to initiate your drug library, it's quite important to have some stable compounds, and stability with salt formation is quite important for the different structures. Yeah? And then also protein interactions and so on is more or less also the hydrophilic, lipophilic interaction in the way, and other properties are really important. Yeah? You see here this curve here for a base calculation, for the acids, a calculation, and then here in green for the amino acid with the calculation in this way. And there you have the isoelectric point where you have the Zwitter ion, but the Zwitter ion in most cases has a low solubility in many cases. So that's quite important, that ionization not always has the best solubility for the different stati and so on. And then I have a long list of compounds. I would like really going through with you with the compounds. I clearly see I will stress you with some boring slides now. Yeah. So for sure. Yeah. And after the third or fourth, you will say, "Oh no, we'll give it." But I'm pretty sure that when you follow the slides, and after 2 or 3 years, you will then get into one of these problems, you will remind this and look it up. And thereby, I will go through it even I'm boring than my own teacher was really in this way. Yeah? So thereby, we can see here some pKa values of amine motifs, yeah? And amine motifs which are highly or very frequently used in drug discovery processes. Yeah? And you can see here ammonia, methylamine, dimethylamine, or benzylamine, and you can see the pKa value of 9.2, 10.6, 10.6, and with the benzylamine only 9.3. Because of the aromatic substituent and the phenylic substituent, it strongly changes this way. And if you then go for aniline, yeah, you see pKa value of 4.6, so much less basic than most people would anticipate it, because of the aromatic structure and the mesomeric structure it can form into the different ways, the -M effect in combination. Yeah? And if you go here for diphenylamine, yeah, you can see 0.8. In fact, this list here, the 0.8 was localized here at this position because hydrophilicity, and these compounds are ordered according to the area, and I had to put it into this list to meet the requirements of this slide here. Yeah? But there you can easily see in this way. And then you can see here tertiary amine, but you can also see here this primary with the here, and otherwise a phenylethyl moiety here. So nomenclature makes it's a phenylpropyl structure, but you can see 9.9, which is totally different to this structure in this element. So you see there are strong difference in basicity depending on the substitution pattern. And here we are just with this kind of biogenic amines, we can go into some heterocycles in which the amine is directly included. So we go here for the piperidine with a 6-membered ring system, or 5 or 7-membered ring system, the azepines or other structure, or even with a double bound here. And you can see here with the piperazine we have 11.1, and the piperazine with the methylation and then double bound 11.6. So slight variations, but this slight variation changes the protonation on the target. Because we're not only discussing pH 7.4, the target in the lipophilic area could have also different protonation, deprotonation status in this way, and thereby change the different areas. Yeah? Yeah? Or the tetrahydroisoquinoline moiety 9.1, or some 5 or 6-membered heterocycles in this way. And you can see here 9.4 with this tetrahydro piperazine, and then only the methylation status here changed it to 11.6. Yeah? So strong changes with small structural variations, and that gives the possibility for fine-tuning in medicine chemistry. So with this fine-tuning, you have an excellent possibility to get into further structure. You see this picture of this automat there. It's not just for fun there. This is a titration automat. So when you have some compounds, and this is an automatic titration, and with the automatic titration, you can give your compound directly inside, and then it gives you the titration curve, and you can calculate the pKa value directly out of this curve. So it's a not too expensive instrumentation. You can do it on your own by potentiometric titration and so on, but this is an automatic procedure, and the pharmaceutical industry, most of them have something like this once to screen all the compounds for pKa values for the physicochemical properties in this way. You see, fluorination is also very important because fluoro is mostly stable against cytochrome P150, and then we do have different possibilities for fluorination, for stability, and so on. And when we have the amine, you can see also that with the different position of the fluoro substitution here, fluoroethylamine, fluoropropylamine, trifluoromethylethylamine, not the correct nomenclature, but just to explain the correct structural elements, you see that we are changing from pKa 9.0, 9.9 to 8.7. So the sigma effects are quite important, although you do not have the direct connection of fluoro to the amino moiety. When I was a PhD student and I learned this for the first time, I was extremely surprised that this kind of interaction really existing, because I was told that sigma interaction only goes for one sigma binding and so on, and that must be a direct effect on this moiety, and this fluoro does not have any effect on this structural effect, which is totally wrong for medicine chemistry. So thereby this minus inductive effect strongly consists on different sigma binding properties on the different moieties, and you can see here, even with a trifluoromethyl group or difluoromethyl group, you can change here the basicity on your will, more or less, in the way by different positioning in this way. And that's what I'm mentioning. When you see it later on and you have to optimize your amine moiety, you go for different substitution, you go for different fluorination and so on, and not really directly on this moiety in this way. One point I want to see also, that some of these moiety, especially the difluoromethyl moiety, they are accessible for hydrolyzation. So some kinds of the structure can get hydrolyzed, and they are not as stable as they look like other fluoro compounds. Yeah? So that must be aware that there are differences or difficulties into this different area. Yeah? Now we have seen basic heterocycles, and we have seen fluoro substitution. And now we are seeing here fluoro substitution on basic heterocycles in this way. Yeah? So you see that the combination of the different structure gives even stronger effect. You see the piperidine here with a trifluoromethyl moiety here, or trifluoroethyl moiety 6.8, whereas monofluorination here gives 9.3. Yeah? Monofluorination in the para position or para-like position gives 9.4. Yeah? Or I'm not sure if I have also the defluoro structure. Not with this one, but I have here with the meta-like structure, the defluoro positioning, 7.4 in comparison to 9.3 with a different structure. Yeah? So you see that not only monofluorination or the defluorination or desubstitution could have great influence. And when you then go, for example, for smaller molecules, then you go for compounds which do not show really any real basicity anymore in the structure when you have this small acetidine molecules in the different structural elements. Yeah? Just to show the flexibility you have. And my most important point is here, like this structure here, where you have the benzyl moiety in substitution and the fluoro substitution. Then you change here the basicity for 8.8, and you make the comparison here for 9.3. Then the benzyl moiety gives the additional properties. Do you see any difficulties you have with the desubstituted structure with the fluoro and the benzyl substitution here with the lowest compound here with the 8.8 substitution? Any difficulties? You have two stereocenters here. Yeah? Yeah? So stereochemistry is an important factor, and then at least you have 4 different compounds. Yeah? So when you do not determine the stereocenter, you have 4 different compounds, and that makes it much more difficult. And that's the reason why so many people go for defluorination here, because with the defluorination you avoid the stereocenter in any case in this way. So this makes it easier to happen and go for different aspects. So sometimes the defluorination in meta or in para-like position, that is more preferred than the desubstitution on different positions because of the stereochemistry in this aspect. And thereby, don't make the things complicated at the beginning. If you need the stereochemistry later, you can introduce it. As screening procedures, I think you should avoid some of these examples quite clearly in the different aspects in this way. Yeah? And you see, I'm not really stopping by fluoro substitution. I take oxygen as a more common substituent in the different ways. And you see, we are continuing the same story for the substitution pattern with the amine moiety, here the tertiary amine, and with oxygen here, the methyl ether, or in a smaller side chain, as a hydrogen moiety here, and going from 9.4 to 9.0, and maintaining the long chain here for a butyl chain with an acetyl ester for 9.4 this way. Yeah? And having then the lower one where we have the propyl with 9.0. And you can see here that even here ethyl change, 9.0, but alcohol versus ether, or we have then here the propyl alcohol with a 9.3. What we are missing here in the pKa determination is, for example, that for sure the ether here is much more lipophilic than the alcohol because of hydrogen bonding. So then the log D determination would be important to get in combination with the pKa determination to determine the ratio between hydrophilicity, lipophilicity, protonation status, unprotonated status in the different ways. So that makes it then more and more complicated in which way you combine the data you have, and that hydrophilicity is not only hydrophilicity or lipophilicity, is combined with some other factors in the way, and here protonation status in the way. And as I've shown before, going into heterocycles, then also here heterocycles with or without oxygen into the different areas, with the morpholine moiety here 7.4, with the hydroxy piperidine here 9.2. So you see, although you have a 6-membered ring system with a tertiary or secondary amine structure, you do have strong variations, 2 log D values or 2 pKa values here in the variation for the structure. And even then you can go here for ring enlargement or ring substitution with different structure, and being then at 9.0 or 9.1, 8.8 in the different areas. Yeah? What gives then great influence on the different structures are here the structure with the keto functionality, quite often used for spin labeling of the different structure, where you use some NMR experiments and can use spin labels with the stable NO or N structure for radical structure elements, and then you go here for 7.9 in the different structure. Yeah? When we here see the tetrahydro oxazole moiety we are having here, or the oxazolidine, 7.3, Do you see here any problems with the structure? Is it stable or not? This is in a half amino structure. So it is connected, and if it's then here protonated, then it liberates here the imine. The imine is hydrolyzed, and this ring system is not stable against chemical hydrolysis and so on. And then it forms, at the end, it forms from this structural element formaldehyde, and that is then the element which is then used for this. And formaldehyde is toxic, so. Yeah, so thereby, you see that there are also some elements which may be useful, but also give some problems in metabolic pathways and different areas. That's the reason why we are always here on one point of this structural element, but I try to discuss some other points in connection to the different areas in this way. Yeah? So then aromatic substitution with the amines, I said I'm getting bored with this different elements in the different structure with some amides structures with smaller or longer chains, or sulfonamide structure with a primary or with a tertiary amine structure between 9.9 and 8.5 in between the different structure. So just for being clear, I took these slides from DrugHunter.com. So and they are freely available. You just have to register. You do not have to pay for this. So they are quite useful for the structure. And I cite also my references here. So that's also important for variability in this way. We just came into sulfur-containing compound in this way with the sulfonamides. And here you see some thioether sulfoxides and also other thioethers. And depending on the chain length here, 10.5, 10.4. And we have here with a shorter length 9.9, 9.0, and even more hydrophilic. So pKa values strongly vary with the different structural elements in this way. And you see that the influence of the sulfur getting less with the longer alkyl chain, what we would have expected more or less in this way. So they're not always the unexpected. Sometimes it's really the expected effects in this way, but you can vary the different possibilities in this way. But also here, sulfoxides are kind for metabolic instability, and thioethers can be oxidized when you have then some active metabolites for the sulfoxide and sulfones, and there are different possibilities for oxidation, so different possibilities for active metabolites with a different pharmacological profile within the structures. Yeah? Here we have some carbonyl compounds with a nitrogen compounds, some amides and some benzylamides in this way. And you see here within ester moiety 10.4, very small variation. The same with the smaller variation here, 7.6, where you have some 2 or 3 log units differences in the pKa values for the glycine ester in the different structures in the way. But you also see as a ester substitute or variation, the cyano compound also very strongly in the same direction for the same area. And once more, when we have the cyclic structure or here some phenylic structure, phenyl substitution here, phenylamide 7.7 in comparison to 7.6 what we have here. So you see that you can strongly vary by different variations, and you do not only have to vary then the substitution on the aromatic phenylic moiety or so on. So there are much more possibilities in which we can go on for the different elements in this way. I have to check, I think this is my last slide for this, or before my last slide for the drug discovery process here, with the amino acids here. Yeah? And you see here amino acids with the guanidine moiety, with the fluoro substitution in the pyrrolidine moiety, and also some basic structure with or without the different structural elements. You see here, especially with the pyrrolidine, 10.6, fluoropyrrolidine, 8.6. So just one fluoro in the correct position gives some other effects. But once more, with the amino acids, you always have, instead of the glycine derivatives, you always have the stereochemistry, and with the monofluoro substitution, you have once more the stereochemistry problems in this way. Yeah? That's the reason why they're using here the hydroxy group, also easier for preparation, I must say. So that can be done in the different methods in the way. But meanwhile, some of these elements can be bought, so they can be easily added to the different structures. Yeah? And this is not only true for smaller molecules, for amino acid, fluorination also here with the larger molecules here. You can see here what I've shown before with the amine motifs in the different structures, a little bit differently ordered in this way. Here you can see the translocation of the fluorobenzyl substitution, yeah? Translocation. And here you see the cis location for the structure. You see the difference between 8.8 and 9.3. So stereochemistry does make a sense here to just from the physicochemical properties in which way they really interact into the different ways. So thereby, just check out when you want to go into stereochemistry or not. My recommendation is stay away from it as long as possible. These fluoro compounds, some of them you can really buy, but they are highly expensive. So because they spend a large work to prepare this, but nevertheless it's much cheaper to buy these compounds than to prepare them on your own. Because if you look for all of the ingredients or your wrong experiments you're performing this, I recommend buy this material instead of synthesizing it on our own. So then, now we found, we finished with the pKa values more or less, yeah? Not really, because we are going here into the heterocycle list, and you see here pyrazosine, piperidine, and other heterocycles, so blue, the nitrogen-containing heterocycles, the oxygen-containing, sulfur-containing, or both containing heterocycles in the different structures. They show really in a tremendous way, really. Yeah? And that's here also shown that it's a large application. Now, some problem with this. Yeah? And I have shown here one compound here. It's not caffeine, but a related structure with the benzimidazole moiety, or larger fused cycles where you have this pyrimidine moiety here in the different structure. And then you can see that with this element here, you have an amidine moiety, a guanidine moiety. And what you have here is also a guanidine moiety with a methylation status in this way. And then, is this compound monoprotonated or deprotonated? And if it's monoprotonated, at which position is it monoprotonated? On the 6-membered guanidine or the 5-membered guanidine? And even when we declare that the 5-membered guanidine is the position for protonation, in which position of the nitrogen is it protonated in this way? So when you go there into details with some of the compounds, it's getting more and more complicated. Thereby, I choose the simple amines as nice examples for explanation. And here you can see with the amidine here, or this guanidine, normally you have the protonation status where monoprotonation status would be here at this position, because that could be done here. This could be protonated here, but it could also be deprotonated into this structure. So there are possibilities in which way the sp2 nitrogen could be protonated into the different structure. The calculation here is a little bit difficult because this guanidine here is an acylated guanidine. So basicity is strongly reduced with acylation. On the other hand, this guanidine here is by the double bonding structure here, it's a phenylic or allylic, also acylated guanidine, which introduces the mesomeric structure or the mesomeric influence up to this guanidine structure. So without careful inspection, it's for me not possible to say which position would be first protonated, second protonated, or which has which acidity or basicity for the different structural elements. So then you see when you get into a mesomeric structure, you have different protonation status and so on, and then different possibilities for protonation. At this moment, it's just a theoretical question in this way. If you go then into the receptor interaction, then it makes really sense if this protonation status on this structure is interacting with an acidic structure here, then it seems to be essential to be protonated here. But if it should be protonated here, we must have a different structural element for the different elements. And then we must see in which way our target introduces the protonation of our compound. If we have the carboxylic acid here, for example, asparaginic acid, then this asparaginic acid can be transformed into, or our compound, our guanidine, our acylguanidine can go inside, and then the proton can be transferred from the asparagine to the guanidine and make a salt bridge. Then the salt is formed directly into the receptor binding pocket in this way, and then it must be exactly here for the interaction of the different structures in this way. So thereby, some kind of induced fit or induced protonation status also must be taken into account when you consider the binding properties of the different structures. So thereby you see it's just determining the pKa values is not always sufficient. Sometimes you have to go into the details with the receptor interaction to make a more clarifying interaction of these different structures into this way. Yeah? Here's also one of these slides where the methods for the characterization of the different properties formed. Here for the pKa value, solubility in combination with pKa value, permeability with the pKa value, metabolism, protein binding, and excretion in combination with the pKa values for the different structure. Also, once more, I believe with Drug Hunter, you will find this structure, or with the Cambridge. It could also be with the Cambridge Hunter side there in combination with this. Yeah? There you see that there are some possibilities in which way you can determine these different structures in a certain way for this. We have now determined lipophilicity, and then we determine the efficacy of our compound or the inhibitory properties in this way. And sometimes this term of lipophilicity is then being calculated or being taken into account with the activity, the biological activity of the compound, taken as an IC50 value or a Ki value for the different compounds. And then you have so-called ligand efficacy. And the ligand efficacy is taken into account the ligand lipophilicity and the efficacy for inhibitory compounds. So when you have different compounds and you describe the ligand efficacy parameter, then you can describe in which way lipophilicity enhances your inhibitory activity. And they are just by increasing the lipophilicity, not by increasing interaction, just by increasing lipophilicity. And normally you should avoid such behavior because you should increase the interaction with the target and not increase the lipophilicity, because this reduces your hydrophilicity, you reduce your solubility, reduces your metabolic stability in the different way. Yeah? And then there's also a possibility where you take this into account for the ligand efficacy, where you have then the LLE, when you have then this calculated together as a log P or log D value into this way. So there are possibility where you put these things together and thereby make a better calculation, because I strongly believe you already see that we just have calculated lipophilicity on log D or log P values, and we have just taken into account pKa values. And I'm pretty sure that some brains are turning around a little bit already. When we're now taking into account IC50 or pKa values, it's the next round in your head, in your brain going around. And thereby, these calculations give you some rough estimation in which way these are connected to each other, and you can then find some structure-property relationships in this way in comparison to the activities. Yeah? To be full, I have taken the references where most of these data are taken from in this way, but I can easily go on. I'm not sure how much time I have left. 50 minutes? Yeah, I take everything I have. Sorry for you. I can talk the whole week through without any. Just have to take some water and something in between. Yeah? Here, now we go back for the drug development program, and where we find the pharmacological target, the lead structure, make the pharmacophore identification, optimization, drug candidate, and so on and so on. We have discussed this already several times. Yeah? Although we are short here in time, we have discussed this already. But the main purpose here is that together, pharmacological target and lead structure must be together in this way. So we could not have a lead structure without a target, and we could not have a target without a lead structure in this way, or even more difficult to go into details with the different aspects in this way. And at the end, we have calculated the duration and the quantity of effect and so on. But you see, for a drug, it is important to have a good quality, to have a good efficacy, and to have a good safety. Yeah? So for the drug to going into market, it is then safety very important, efficacy important, and then also quality of the drug is really important. And safety is really an estimated thing. So when you get a better drug, which has a lower toxicity problem, then this drug is preferred because it's a much higher benefit-risk ratio and so on. We have discussed some problems on the pain structures. And pain structure, you can see here the Calotte model of morphine, yeah? The structure of morphine. And you can see here the endogenous ligand, which is methionine or the metencaphaline with a different structure. You can see here slightly tyrosine and phenylalanine in the structure, and these are combined by some glycine molecules, and this methionine with the sulfur does not really reflect so much to the binding to the morphine receptor, but you see here that it's a morphine, it's a not endogenous ligand, it's a ligand by the plants. We've already seen by Papaver somniferum in this way, and this gives here this interaction which is slightly reflected by tyrosine and phenylalanine. And now we can go, for example, for naloxone, and you see here the same interaction area, the same pharmacophore model with the same structure, so it can also bind to the same receptor, and it can have the similar profile in this way. But similar profile just for binding, because this naloxone, you can see here, has here a lipophilic tail in this way. In fact, this is an allyl tail in the way because it's called N-allyl morphine in the way. That's or oxymorphine. That's where the name naloxone came from N-allyl. And this lipophilic chains make this from a full agonist to a partial agonist. And this partial agonist inhibits the structure, and thereby it's an inhibitor of the full agonist in this way, and thereby you can see here pharmacophore development is very nice, and with the pharmacophore development, sometimes by increasing the structure, you increase the antagonist behavior. Yeah? Thereby, most of the agonists are quite small in this way and the different structure. And you can see some displacement of the different structure could then lead to some new elements in this way, and this is we call bioesteric replacement. Yeah? Bioesteric replacement is, for example, methyl against amino or alcohol moiety, or bromo against isopropyl, because they have, from a classical point of view, they have almost more or less the same size. Yeah? So this classical device, not concerning orientation, not concerning electronical properties. So what you're finding here is just that these elements, monobinding, dual binding, or triple binding elements, they just have about the same size. So from a alkane, you can easily go to an imine moiety or something like this, because they have almost the same size in the different elements and so on. If we go then for electronic properties, then we cannot stay with this. One point I missed here. Quite often, Bioesteric replacement is done with mini compounds, hydrogen against deuterium. Very famous possibility to overcome patents, because patents going for the compound, and then you exchange a few of the hydrogen atoms against deuterium, and then you can have a new patent because you have new compounds in this way, and you just show that these are compounds. So very easy example to overcome some patents because they are not included maybe into this area. Just to show that bioesteric replacement is quite often done because of the patent overcoming from other structures in this way. Then the non-classical moieties here, going for the carbonyl moiety, there we have the hydrogen bonding acceptor properties, but also the partial negative elements in this way. And then you can see here that the sulfoxide, sulfone, or sulfonamide, or amide, or even the cyano compound has some similar properties. And even the dicyanoethyl moiety here, highly polar compound with some partially negative electronic properties at the end, displays the carbonyl moiety. And thereby you see, when you have a carbonyl moiety in your compound and make some bioesteric replacement, you come to new compounds, new fragments, new elements, new compounds for patenting. So that's quite often the case where you go for it. Another important point is with the carboxylic acid here. Carboxylic acid, where you go for hydroxylamines or phosphoamides, or for triazoles. These are quite important with the triazoles, and these are the most used bioesteric replacement for the carboxylic acid, because you have the NH acidity of this compound. You have very much the free electronic pairs for hydrogen bonding acceptor properties with this nitrogen here. And if it's then deprotonated, then the negatively charged is mesomerized, stabilized during the different structures in this way. So, and it has a higher metabolic stability. So often when you have a carboxylic acid and you want to exchange it, the tetrazole is a very good step for non-classical bioesteric replacement within the structure. The thiourea is quite often used, and you find the thiourea in many of your structure. Unfortunately, thiourea has some toxic properties. It quite often induces agranulocytosis, and this agranulocytosis is a blood disease which can have lethal department. Thereby, the thiourea moiety is mostly exchanged by some elements, and then you can hear the cyanoguanidine or nitroethane diamine, what you have here, and in all of these elements you have a highly polar element and a non-basic element. So basicity of these amines moiety are reduced by the ethylene nitro group. Basicity of these amine moieties by the imine cyano moiety reduced in this way. So high polarity but low basicity in this way, and thereby you maintain the electronic properties and you get rid of the other elements in this way. Or for a catechol element, because we are working with the dopaminergic ligands with a catechol structure, we quite often have a substitution of the catechol structure. We have the binding of the both hydroxy moiety with the benzimidazole, with the hydroxyketo moiety with different heterocyclic elements in this way, and different possibilities here for interaction. Unfortunately, most of these structures have a higher metabolic instability, but they are more stable than the catechol, which is highly redox sensitive in this way because of the redox activity in this way, getting into quinoamines or hydroquinone structures in the different elements and this more. So I just want to explain a very recent example where some bioesteric replacement was successful. And you see here that this morpholine structure with the piperazine moiety was highly effective as an anti-cancer compound. The mTOR is a mammalian target of rapamycin is a regulating enzyme or regulating complex for cell proliferation, and this compound has a quite high affinity with the PIC50 value in a nanomolar concentration range, but you see it's for other compounds or its solubility was quite low, and even log P value was only moderate in a different way. And then this could be slightly modified into a double bonding instead of this structure into the alkyl chain, or introducing a cyclopropyl moiety here. And then this moiety here maintains the activity, got higher stability concerning other structure, maintaining the good lipophilicity, and got higher solubility even more with a different structure. Yeah? So you see that there are some possibility where you would not always expect the different structures, but you see that also the synthesis route going from the boronated structure to the cyclopropyl moiety to the borofluoryl structure, and then make a Suzuki coupling here for the heterozygous structure is quite much more work than it has been before, but going into much more effectivity for the different structure in this way. You have not always go into this highly sophisticated modifications. The last example I really want to show for the bioesteric replacement is that you get here a cinnamoic acid derivative or a chalcone derivative in which the dichlorophenol compound shows bactericide properties in this way, and but low solubility. And then we exchange the hydroxyl group against the carboxylic acid and chloro against bromo. We even get better activity because we have higher solubility, but we're changing the properties from bactericide to bacteriostatic. So it stops the growth, but it does not it is not toxic to the bacteria by himself. No? The same is true when we make the exchange by the trifluoro. There are also some possibility to trifluoromethyl group, other exchange, and trifluoromethyl group is much more stable than the other groups in this area. So you see minimal inhibitory concentration is reduced with both of the compounds. So the bioesteric replacement is not only bioesteric, it gives some improvement with the compounds, and we can get into further details. Yeah? And thereby I hope I have shown to you that the physicochemical characterization is quite important, and that with some small variations you can easily go into variations into your scaffolds into this way, and that you can easily take into account bioesteric replacement. So if somebody other has a lead structure, you can make some bioesteric replacement and check if this is there. The only thing I want here to mention with the bioesteric replacement is in most cases oriented to the target. So bioesteric structure on one target must not be the same affinity, the same replacement on another target. Because bioesteric means recognizing as a very similar structure having the same function. And not all targets make this recognition. So bioesterism depends on your target in this way. So these are some general rules which are very often applicable, but in most cases it depends on the target in which way you go through with this. Anyway, thank you very much for your attention.
You all can ask your questions now.
Maybe one point before you go or before you leave, there's one point I will continue this topic for bioesteric replacement or with similarity or differences on Thursday. Tomorrow will be mostly ATME properties, but then we proceed with this topic in this way because the drug optimization, lead optimization concern thereby the similarity and is more related to this one.
Yeah, good evening, sir. Sir, I have one question, like the slide of solubility.
Yeah.
Yeah, so you are using DMSO in buffer. So why DMSO?
No. we're making solubility in DMSO. Yeah. So very small amount, the minimal amount of DMSO is used.
Preparation for that molecule.
Because we want not to test the dissolution factor, we want to test solubility. This when you have the compound, and then it goes into solution.
Okay.
So that is also a kinetic factor where the dissolution speed is strongly influencing than when you're measuring. Yeah? And that means you have dissolution is different and solubility different to liquidity.
I got it, sir.
Yeah.
But my question is like, why this like we are using organic molecule for like bio-tracking and biosensors applications like some enzyme sensors in the human body. So I have gone through many research papers. So they are preparing stock solution in 8:2 ratio of buffer and DMSO in most of the papers, and they are not saying why they are using DMSO.
Okay. Because any of the compounds you're using, they're not soluble in the buffer on its own.
Yeah,
so,
I got it, sir. But in dioxin also it is possible.
So you cannot make the screening. So you add, in most cases, or you make a standard solution in DMSO. Yeah. Because DMSO is a very universal solvent in the way, lipophilic, hydrophilic, different elements in this way. So most compounds are soluble in DMSO, and then you make your stock solution in DMSO. And then you dissolute your compound step by step in different buffer, and then you have a known concentration. Maybe with DMSO, with the remaining DMSO, which is unfortunately, but you maintain then the exact concentration of your compound. And then you can tell if your compound is active at a nanomolar or a micromolar concentration range, because you have the exact concentration in combination with the DMSO.
We have to optimize that one.
Yeah. In fact, we are when we are using different concentration status with our compound, and we are using different stock solutions, sometimes we have different participation of DMSO. So one compound has 10% DMSO and the other has 30% DMSO. And then we equalize this, we add to the 10%, we add 20% DMSO to have 30% with all of our stock solution, that we can compare the data, that we do not have the influence of DMSO, and that could be avoided by this.
Sir, like if my organic molecule is soluble in dioxin or chloroform, so can we make stock solution with the chloroform aqueous solution?
No, that's not possible because chloroform is not soluble.
In case of dioxin, just leave.
Yes, but even with dioxin, you have a limited solubility in some buffers. Yeah.
Okay.
DMSO, it's a unlimited solubility.
So that's why they are using DMSO.
That's the reason.
Got it, sir. One more question, sir. In the affinity table, you have proposed attaching different subgroups, the gaining affinity and so what is that affinity? It's related to biological activity or something else?
That depends on the target, what you're screening for. So what I suggested was the fragment screening by fluoro NMR screening. That is just measuring the interaction between fluorinated compound and the interaction. Your compound has some fluoro ligand signal in combination with the protein target, so it gives some Schirming factor with the, I don't know the English name. In German it's Abschirmung for the fluoro NMR signal when it's bound. Yeah, okay. And then you give your non-fluorinated
binding power, we can say, like affinity is related to receptor and ligand binding.
Yes, it's just by non-covalent binding, just by interaction, by reversal binding, they show a reduction of the fluoro signal in this way, and then you can measure the signal. And then you put your compound on this, and if your compound is reducing the signal, then or increasing, then it has also some interaction. And in many cases, you're not using one fragment. You're using already 10 fragments simultaneously, and then give this to your compound, and then you have 10 fragments screening simultaneously. If there's some change, then you go into detail and you check which of the fragment was causing this factor. So it makes it a little bit cheaper because you're always using some protein and your fluorine ligand.
It's not related to cell viability thing, no?
What?
Actually, I have a case, like piperazine moiety, the 6-membered NN ring. Sir, I have one molecule that is highly bioactive, but I attached piperazine in that particular synergic system. The bioactivity is almost nullified.
Okay. Yeah.
So cell viability is 95%. So I'm figuring out the reason behind that. So I got that in the slide that piperazine is reducing 100% loss is there. So that's why I'm asking.
It could be the case that you're losing your affinity because of protein binding, for example, that you have in your another protein, and this protein binds a compound.
Yes, sir. I have a good docking score. Simulation is also.
Yes, but with docking, you're just checking the proteins of interest. Maybe you have some other
I have
albumina.
used some tools also for calculating IC50 and cell line, which is more suitable. Everything is very fine, but in vivo studies, it's not working. So
I can clearly say that we have developed some COX inhibitors, and these COX inhibitors were excellent compounds in the enzyme screenings, and when we went into cell screenings, only half of them were active. But they were still, but at least half of them were active in almost maybe 1 log unit less, most of them, and they still showed some activity because there were some interaction with the membrane because we're working on lipids and they are stuck in the lipid membrane, and then they were stuck in the membrane, were not accessible for the enzyme in this way. So that could be easily clarified, whereas some other compounds were not stuck to the membrane so nicely, and then we can have the direct interaction. That means some unspecific interaction mostly are responsible for this deterioration. Sometimes also some additional enzyme activity which is in your other cell system and catabolizing your compound or making other metabolites could be the case. It's different cell lines make different effects.
Got it. Thank you, sir.
Do we have any more questions?
So I have questions on that. But did you understand the pK value discussions what he made? That's question to you actually. Did you understand how it is correlated? Because I feel those slides are really important on that. The question what I'm going to ask, so does it correlate with the pK value, with the deuterionic structure of that amino acids things? Like, because that also matters there.
With the amino acids, you can see that pKa values are quite important because you have the acidic and the basic elements simultaneously there. And there are also some amino acids which are basic or which are acidic. They have an additional amino or an additional carboxylic acid on the structure or basic structure like a guanidine or something like this. So they strongly influence the different properties. For example, if you have an arginine, then the protonation is on the guanidine moiety and not on the alpha amino structure on this way, and that gives us a very unusual amino acid. The same is true with pyrrolidine, where you have the amino group in the ring structure enclosed in the way, and you have a secondary amine in the structure. And this is strongly reflected by some protein structure. For example, when you have a proline-rich structure in a protein, it's mostly concerned as a turnaround structure because the beta helix structure turns into alpha fold structure into the different structure by a lot of proline structures. So the amino acid and the amino acid sequences strongly influence the protein structure in this way, and thereby this makes really clear influence on the whole structure and the binding properties. And you can see here. I may also remind here, I'm in the advisory board of a new company here, and they are developing some Alzheimer compounds. And these Alzheimer compounds are also small proteins, but these proteins only exist of the D amino acids. So instead of the L amino acids, they have the other isomeric structure, the the D amino acid structure, and then they have a short sequence of this, and this sequence is much more stable because there are no enzymes available to cleave D amino acids. They are for the L amino acids in the body, so they are stable for quite a long time. And they are the mirror elements, so they bind good to some other targets in this way. So even by some change in the stereochemistry there, you can change the metabolic stability because of the enzymatic cleavability of proteins and so on. And thereby, especially with amino acids, you can play around very much with the very small sophisticated variations and so on. So that's I think one of my favorite, but one of my problematic areas, of both cases.
And how difficult it is to titrate? What are the instruments you have shown? Like, is it like very regular?
Titration is very easy if you have enough compound. Yeah? So with our undergraduate students, we make some titration for pKa determination, and they do it for learning to learn electrochemistry on potentiometer structure. But then they have a few hundred milligrams of the compound. Yeah? If you go into a smaller amount, if you have only a few milligrams of your compound, it's getting much more complicated. That's why they use this titration automat, because with the potentiometer, you have to put your potentiometer in your dilution. And when you only have half a milliliter, it's difficult to put your potentiometer just from a technical point.
Yes,
you have to buy new equipment for this,
yes.
And when you already have to buy new equipment for this, you can buy directly this automat for this. So that's then the best and cheapest way, because they are reducing the solvent necessity. They do have some specialty, but they also have consumables.
Yeah. So in every cases, the reagent systems are like same for all the pKa?
They could be the same, but it depends on the buffer. The buffer depends on your screening system. So most of the screening system contain of potassium phosphate buffer. Yeah? But for example, we have also tested for calcium channel properties, some compounds, and thereby our buffer needs to be fully calcium free. And then we have to test our lipophilicity or basicity on calcium-free buffer, which is some other ingredients, and then we measure something else in this way. So, and you must measure in water and not in something else. So water with something you're using. Yeah?
So do you have any additional questions?
Hello, sir. Sir, you have discussed here about the effect of fluorine, like whenever they are using fluorine, that lipophilicity, bioavailability, those are increasing. So did you came across any compound where because of the presence of fluorine, that toxicity increases, like it plays exact opposite role?
Increases lipophilicity or reduces lipophilicity? What do you mean?
Because of fluorine, like presence of fluorine may increase the lipophilicity, biological activity, like binding affinity, those things increases in presence of fluorine.
Yeah.
Like did you came across any molecule where it plays the negative role? Like it is not mandatory that presence of fluorine will increase the biological activity.
Yeah. The substituent effect of fluorine in many compounds is well known in aromatics substitution. So the change for hydrogen to fluoro in phenyl group, for example, in a paraphenyl group, is quite often performed to increase the metabolic stability. Yeah? Because then it is could not be hydrolyzed, and the lipophilicity more or less stays the same. But if you exchange, for example, a phenyl group against the fluoro group, then you increase hydrophilicity by the fluoro substitution. It depends what you're looking for the exchange. If you're looking for hydrogen exchange or for phenyl exchange. So there are different possibilities. And sometimes the fluoro group is sufficient and can replace a full phenyl group if it's just for steric effect or for the same is true sometimes with the chloro. Chloro has a larger steric demand, and with this larger steric demand, the non-binding electron pairs are more outside of this area, and in some cases, the people claim a magic chloro effect. the magic fluoro effect is by the non-binding electron pairs, which makes, instead of chloro substitution, which is normally getting more hydrophilic, getting better hydrophilic interaction with the target. So there are some other possibilities in the interaction, and there are some effects which are done. Then you have to take, when you go, for example, into not para or meta, but into auto position, when you make some substitution in the auto position, sometimes your ring orientation change into this one just by auto substitution. So you change your whole orientation in your ring by the auto substitution, and it's not because of the electronic, but of the steric effect of your substituent, and thereby you can get some other variation, like this, or even with some stereochemistry, some axial stereochemistry could also be done. So there are different possibilities. Not always one rule you can apply for everything. That's unfortunately not the case. Yeah, but the better you know the interaction with your target, the better you can modify it. Yeah? So if you have, for example, an X-ray structure or a cryoelectronic structure with your interaction of your ligand with your target, that would be excellent. Yeah? Then you know the binding pocket, you know the interaction partners, and then you may do some optimization with this. Structure-oriented optimization.
Sir, in the 2023, in the year 2023, number of drugs and some biosimilars have been approved. So, how the drugs and biosimilars are differentiated? Yeah,
biosimilars are different to the small molecules because the general structure of the biosimilars are similar, what the name says. The main problem with biosimilars is that maybe you have the same protein sequence, so you have a large biological structure, but there are many post-translational processes in the biologicals. And with the post-translational processes, you got glucuronidation, galactosination, palmitylation, and so on. And depending on your process of the preparation, it is different. And thereby, they are not bioidentical, they are biosimilars. And to my mind, the biosimilars are well treated. I would be more careful with the biosimilars because they're not the same compounds, because they have other post-translational processes in this way, although they have the same structure in the way. And thereby, they behave a little bit different. And you can see it with small molecules where they have different preparation procedures and so on. The NIH has some procedure when you got a drug unavailable into market or got into rare delivery into market. It was the case with the GLP-1 ligands, and because I mentioned it only because it's so popular at the moment. And then some drug marketing companies could prepare this compound or are allowed to prepare the compound on their own and then give it as a substitute. So as a similar structure or same structure with the element. And in fact, there were, I think, 6 or 8 deaths in the United States only caused by GLP ligands, and all of them were contributed by these non-legalized structure element, which has some changes or some not the biological screening in this way. With the biosimilars, they just have to show that they have a related effect and they have the same pharmacokinetics. So that's sufficient for biosimilars, and I think that's a very simple solution for different compounds, but the money spent for biosimilars in comparison to the original products was one of the driving force, to my mind, to make something like this. But I think they call it biosimilars because they are not bioidenticals. Yeah? And that's a problem with the compounds. So if you want to change from one biosimilar to another with the same patient for the same diagnosis and so on, you may get into problems.
Yes, sir. My question is, when we are developing or just theoretically developing some compounds, like we are target some different protein for a, let's say, histamine receptor, and we are designing some compounds. Now we are screening the compounds theoretically, like log P, log S, topological polar surface area, sometimes binding energy. And is this enough to screen the molecules, or we are missing some data for theoretical perspective, I am saying?
Yeah. When we have a lot of compounds prepared by my PhD students and so on, we calculate also log P values and things like this and do some. But we were really unsatisfied by the outcome of this, and we could not make any comparison of the in vitro screening of our compound in comparison to the log P or pKa values calculated in this way. And then we started to make some HPLC-based measuring of the lipophilicity and also of some pKa values, and got some results in many cases different from the calculated ones. So the real experimental data were not confirming the calculated data, and we were taking the experimental data as real, more or less. Yeah? When we have established this, then we could test a larger set of compounds. It is because HPLC can get automatically, but it is also much more costly, and running this through the computer is one day, or even when you compile this, you can do it on a routine and so on, and it costs almost nothing. Yeah? When we do this on an experimental basis, I think one data point costs €1 or €2, yeah? Which is okay, but when you have a titration, you have different data points, you have 6 or 7 data points, and then you have a repetition 3 times, yeah? So you have already 21 data points, so one determination costs about €50. Yeah? So then, and when you then test 50 compounds, it comes together in this way. So thereby, we remain this for the most interesting compounds and take this into account. But on a regular basis, we make the calculation. But for the compound worth doing additional investigations, we prefer the experimental stuff. And then with different pH values, even then, because we also look for different inflammatory processes, we want to see what is the protonation status at pH 6 or pH 7, not only pH 7.4. So we expand our studies there, depending on the target we're looking for.
Thanks.
Yeah. I would be surprised if you're not tired now.
So thank you, Professor Stark, for your excellent lecture. Please join me in thanking him in an official way. And so if there is no more questions, then we can close today's session. We'll be sending email for tomorrow's program. So mostly it will be starting from 10 o'clock and then 2 hours lecture, followed by 1 hour and then lunch. And exactly like after the lunch, we'll be having 2 hours lecture. Okay, so that's it from my side. If you have any kind of queries regarding the timings, because I can understand you are having a very vibrant participants, like most of you are from chemistry background, pharmacy, maybe the medical science. So please don't hesitate to ask any kind of questions, because I can understand maybe you are not understanding the actual chemistry part. So please raise this one. So the main objective to run the course is you should be expert in all this sort of domain. Okay, let it be not only medicinal chemistry, the chemistry aspects also. Yeah. So without chemistry, you cannot do medicinal chemistry. Yeah. Those things has to be clarified. So if you have some other questions also, you can keep in your mind, you can interact with him or us separately after the session as well. But those things should be clarified. You have to volunteer yourselves to make the things done. So please keep in your mind and interact more, ask questions. So that's it from our side.
Maybe also one point from my side. I know that most of you are from the organic chemistry background. Yeah?
Not necessarily.
But many of them.
Yes.
And thereby, I did not focus on the expertise you already have. I focused on the expertise you must get to on this way, and thereby my part for organic chemistry would be very small. And you see a very, very small synthesis route are described, which are fine. But I think this is organic chemistry, how to go to this. Medicine chemistry is what concept had to be applied, and I will focus on the concept. And I can get many things much shorter in this way if we want to discuss these different things. So if I'm fully free to, if you could interrupt me in between and we can discuss in between, and I will not make it longer as you need, but there is good, as I said, I have enough stuff for the whole week to put on you, but that would not be the case. It should be directly transferred to you, yeah? In cerebrum.
Exactly. So the course should be very much concrete. That is needed. Okay. So thank you very much again for joining us. So see you tomorrow. Okay. Thank you.
Thank you very much. We still have some problems here, I think.